Hello, everybody. Welcome. So we are now going to cover the material from Chapter 8. Chapter 7, hopefully you went through that, got your general basic understanding of energetics, how energy can be converted and transferred from one thing to the next, from one form to the next, and then also enzymes, how these biological catalysts allow chemical reactions to happen at rates that are friendly to cellular processes. So chapter 8, we're going to take what we learned in chapter 7 and apply it to an actual process called cellular respiration. So if you're feeling a little bit wobbly still with the information from chapter 7, stop by a Zoom office hour, send me an email, ask questions, try to get that solidified up a little bit before you jump into cellular respiration. So just kind of keep that in mind. We're going to be using what you just learned in 7 here in the mitochondria. So without further ado, I'm going to start with an overview and then review our redox reactions. All right, so you have this diagram in your text kind of at the beginning of the cellular respiration chapter. It's kind of like a map. It's kind of like a map of where we're going. So cellular respiration is a multi-step process. It's not just A to B and then we're done. We have multiple steps, and I'm going to explain why we have all those steps here in a little bit later in this lesson. So the 4 stages or phases, however you want to call it, of cellular respiration, the first is called glycolysis. The second step is called the formation of acetyl-CoA. I just call it the acetyl-CoA step. It doesn't have a cool fancy name. Step 3 is called the citric acid cycle. Sometimes I might call it the Krebs cycle. It's just easier to say than the citric acid cycle. And then the last step is the electron transport chain and chemiosmosis. And my head picture here is actually hiding the fact that chemiosmosis gives us about 34 to 36 ATPs. That's the big energy payoff. So steps 1, 2, and 3, we're getting a little bit of energy. Step 4, that's when we're getting most of our energy. So when we go through this chapter, I'm going to be making some videos, these little mini lessons for these stages: glycolysis, acetyl-CoA step, Krebs cycle, or citric acid cycle, and electron transport. But the even bigger picture than this, like, what's the point? If you remember when you were doing your chapter 4 and writing your cell stories, the mitochondria is the power plant. It's the powerhouse of the cell. It is what generates energy for the cell. And for eukaryotic organisms, plants, animals, fungi, protists, they all have this organelle called mitochondria that's been adapted to be able to take nutrients, these chemicals that can be acquired from the environment like sugars and fats and proteins, take those and break them down in this systemic process to release energy. And the energy is in the form of ATP. So that's our goal. We need to take the potential energy that's stored in our foodstuffs like sugar and carbs and starches and fats. We need to be able to take that potential energy stored in those chemical bonds and convert it into still more potential energy, but in the form of the bonds of our ATP, right? So we need ATP. That's the energy our cells can use. Now we eat glucose because that's what's available on our planet, and all of its, starches and fats, all that kind of stuff. We're going to stick with glucose as our starting point, but it's true for all of the nutrients, those major macromolecules. So that's how our food is supplied on this planet via photosynthesis and the food chain and food webs, but our cells run on ATP. So we need some kind of currency converter. So like when you go travel to a different country, you have to turn your dollars into pesos or francs or yen or wherever you're going. We have to have a mechanism in the cell to convert the energy that's stored in those chemical bonds of carbohydrates and lipids and proteins and turn them into the form of energy that our cells can use, which is ATP. So that's kind of the big picture. And then this is the semi-big picture of the 4 stages of cellular respiration. But before we jump into those, I wanted to do a quick review of that redox reaction. We're actually going to be seeing some of these, and I'll highlight them and point them out as we go through. But remember our friend Leo here. Leo the lion says grr, losing electrons oxidation, gaining electrons reduction, right? And then this is the same slide that we saw in chapter 7, but just to remind you, it is the electrons that are being transferred, usually with hydrogen ions, and we're going to see this in our examples. And again, that big picture idea is glucose loses its electrons, and by losing its electrons, it's changing its complete chemical makeup, and it loses its electrons to become carbon dioxide. It doesn't give its electrons to CO2. CO2 is the result of all the electrons and hydrogens being stripped away, right? We see CO2 doesn't have any hydrogens in it, but glucose has hydrogens. So those hydrogens with electrons are being stripped away. And who are they going to? They get deposited onto oxygen. And then the electrons with those hydrogens gives us water. So we can kind of see the electrons on glucose, get transferred to oxygen with those hydrogens. That's the redox reaction. So glucose gets reduced, or sorry, glucose gets oxidized while oxygen is getting reduced, and water and carbon dioxide are our end results. So we're going to go through those kind of in this stepwise fashion, because I mentioned in the chapter 7 slide that we can't directly transfer electrons from hydrogen onto oxygen without a huge explosion. And in lecture, I talk about the reason why we have to do all of those multiple steps, the 4 stages of cellular respiration, and even within those 4 stages, there's multiple steps within that. We have this picture here, and then the next slide is a video that I would normally show in class, but I can't just due to the software that I have. But we'll talk about that when we get to the next slide. So let's say we have a spontaneous reaction. So this guy is shoving this boulder off of a height of a cliff or something. So this is what we call an exergonic reaction. And here is the negative delta G, the releasing of the energy in one foul swoop. So all of the energy that's released from dropping the boulder from that height to the floor is E, okay? Just this quantitative amount of energy. But what if you were to roll that boulder down steps? He would still have, from the same starting point to the ending point, he would have the same difference in released energy, but at every step, we have a little bit of the energy is released at a time, and if you add all of those up, it's going to equal the same energy release as if you're to drop it all at once. Now, for biological systems, this is really important because we are delicate. Our cells are delicate, and if we have a huge release of this exergonic reaction, this catabolic reaction of breaking glucose down into carbon dioxide and water, oxidizing glucose and reducing oxygen, if you were to just do that reaction all at once, we would not be able to survive. And so this is the next slide. This is the video that I show. So I'm going to link this in part of the helpful link. So make sure you watch it because it's super cool. But see, there it is. But if I click on it, it won't let me play it. So the basic chemical reaction that's happening is the electrons from the hydrogen that's in our glucose molecules, right, the C6H12, and the oxygen, right, glucose plus oxygen. What happens when those electrons get transferred from hydrogen to oxygen to get water, right? There's our watery part of our products. It is a negative delta G of 286 kilojoules per mole. Remember, breaking apart one molecule of glucose was what, like 30 something kilojoules per mole? It is an explosive release of light and heat, or heat and light energy. So if you go watch this video, this teacher, he's doing like a presentation in a gym, it looks like. So these balloons that he has filled up, there's gonna be 3 of them when you watch the video. He's got this candle on this really long stick. He's got protection on. He goes and he puts that candle right up next to these hydrogen-filled balloons, and you'll get to see what happens. Now, we do not want all of those steps. We do not want all of that energy being released in our cells at once, because like I said, we can't survive that as an uncontrolled reaction. So what do we get? We take this explosive release of heat and light energy, that negative 230 something kilojoules, and what we do is we take it and break it down step by step by step. So this is kind of the major role of the electron transport chain. It's the reason why we have 10 steps in glycolysis, the acetyl-CoA step. I think like 8 steps in Krebs cycle, multiple parts of the electron transport chain. We still start with the same things and end with the same things, and we get the same amount of energy, but we don't explode, which is a good thing, right? Okay, so that is our overview, introduction to the stages of cellular respiration, a review of redox reactions, and a lot, hopefully logical, an explanation as to why we have to learn so many steps. All right, I will see you next time.